Click for IB Biology. In the principles of respiration, we're going to be looking at different respiratory substrates and comparing the energy yield that we get with those different substrates. There's a PDF of this presentation down in the description, and also there you're going to find a quiz with answers which you can use to test your understanding of this part of the work. Calorimetry is the name of the experimental process which allows you to determine the energy content, in this case, of different types of food molecule. The actual mechanism results in the complete oxidation, the burning of a sample of a food in oxygen, in pure oxygen, and then determining the energy given off. And you can see here that the unit of measurement is kilojoules. So we've got a similar mass of three contrasting molecules. We've got glucose representing the carbohydrates, protein sample, and fats. And you can see that the carbohydrates and the proteins give roughly the same energy content per 100 grams of a sample. That's 1760 kilojoules in particular. However, notice that fats give considerably more kilojoules than either of the other two respiratory types. Now, we're very conscious that when we discuss respiration, we always begin with the respiration, the metabolism of glucose, but it is possible to use protein and fat in respiration. So let's take a look at why these differences exist. As mentioned, if we respire glucose as our substrate, it's broken down through glycolysis into pyruvate. And at the end of that process, one glucose molecule will generate two acetyl molecules. And these are already in the mitochondria embedding into the Krebs cycle. Protein, usually if we use protein to metabolize, it would be true, I think, to say that you're probably in a state of malnutrition or starvation and that you're breaking down proteins as a source of energy. But first of all, you would do so by using proteases to break down the protein chains into amino acids. And then once we've got the amino acids, we can take off the amino group, which is actually called deamination. And that leaves us with this simple two-carbon structure. Now, there are a variety of these determined by the R group. But essentially what we're getting here is something chemically similar to an acetyl group, or perhaps better, a keto acid. Now, at this point, there are a variety of pathways in which we can then link these molecules into the Krebs cycle. But the energy content is pretty much the same as you would get for glucose. But as previously mentioned and stressed, you will only use proteins under a state of starvation and malnutrition. Lipids, on the other hand, and represented here by fatty acids, are capable in their 100 grams of producing many, many more acetyl groups. If we look at the molecule here, the fatty acid with its carboxylic acid group at this end, what happens is that this chain is broken down to form two carbon structures. So every second carbon, a bond is broken to create lots of two carbon structures, and these will become acetyl groups. And so we have many, many of these feeding into the mitochondria, and as a consequence generating far more ATP, and that's where the large energy content of fats come from, and it's what makes them suitable as energy stores, of course. So whilst the lipids turn out to be a good way of storing energy, let's remember, of course, that for all of the acetals entered, they will require considerable oxygen. And so metabolism, respiration, where the substrate is fatty acids, requires considerable oxidation, and this has an interesting physiological consequence in that it limits the stress level of the exercise that you're able to do. So walking, slow swimming can use up lipids, but intense running would not use lipids because there would be insufficient oxygen available, and the process of this would be. One small item to add is that the breakdown of a fatty acid chain into acetal groups is called beta-oxidation. There are links here back to the playlist and a subscribe button and the channel, which will allow you to look at the other videos in the IB Biology series that we're doing here.